Blackman at UC Berkeley. Today I'm going to be talking about the second half of my dissertation research at Kansas regarding transgenerational plasticity in mimulus in response to wounding. I'm going to kind of give as broad an overview as I can in 13 or 14 minutes. I will kind of have one section focused on the molecular epigenetics, and then another section focused on kind of the relevance of transgenerational plasticity in nature. Also want to say it's awesome seeing more talks on epigenetics in an evolutionary perspective this year. Last time I was here, it really wasn't kind of considered from an evolutionary perspective. It's really cool to see. Okay, so I like to think of evolution as a driving force that leads to selecting for individuals that produce these optimal phenotypes in a given environment. And so what that means is that it selects for individuals that are capable of synthesizing information to produce a good phenotype in their environment. That information can be genetic information, environmental information, or epigenetic information, using that to produce something that's able to survive and reproduce. And so as you can see here, this species that I work on, Mimulus catatus, that those different sources of information can lead to vastly different phenotypes. So what makes this tricky is that the world is not just a single place with one environment. There's variation across the landscape, both spatial variation and temporal variation, or variation in time. And because of that, local adaptation is very important. And usually when people think about local adaptation, I think they mostly think about it as far as space, and that's kind of different sites, different populations will adapt different mean phenotypic values based on the mean environmental variables at those sites. But also there's variation across time, seasons, days, there's obviously lots of different ways of thinking about that. And because of that, there's also local adaptation within these sites that selects for individuals that are able to plastically respond to either their current environment or transmit that information between generations to produce phenotypes that are more likely to be successful in future environments. And the specific timescale and kind of pattern of temporal variation that I'm really interested in, I think is really important for transgenerational inheritance, is this variation that occurs between sequential generations, so intergenerational variation. And in particular, when those are autocorrelated, I think that's really important, and it should lead to situations where transgenerational effects can be beneficial. And so, two examples that are kind of climatic oscillations, as well as these predator-prey cycles. So as far as climatic oscillations go, kind of El Nino is an obvious one. But you see lots of these where the previous year is particularly hot, the next year is also likely to be particularly hot. And here's an example across the US where in white we have areas where there's no interannual autocorrelation for temperature. In green there's minor, moderate, and then blue like highly significant interannual autocorrelation for temperature. As you can see, it's very prevalent across the US where there are these positive autocorrelations. And because of these autocorrelations in climatic factors, that can also lead to autocorrelations in other factors, such as herbivory. And this hasn't really been tested too well yet, but it does seem like predator-prey cycles and herbivores both kind of show these patterns where when one year has high herbivores, the next year is also likely to have higher herbivore density at that site. And when that's the case, we have high autocorrelations, and it becomes beneficial to be able to pass on information between generations. So if I was a plant and I was chewed on, not only might it make sense for me to produce more defenses, but also might it make sense for me to pass on information so the next generation produces higher levels of defense. So the system I work on is Mimulus catatus. And yes, in this system, when the parents are wounded, the offspring produce more trichomes. The first part of my dissertation work, which I'm not going to talk about here, we showed that differential trichome production is associated with differential gene expression. And some of these genes we think are probably related to this production of trichomes, as well as other genes related to kind of secondary metabolites. And so today we're going to talk about kind of what's causing this differential expression. Do we see that there's inheritance of these epigenetic modifications? and then do these differential epigenetic modifications associate with the genes that we previously found to be differentially expressed. And then I'm going to go quickly into some evolutionary implications of this. Do we find that there's variation across lines for this response? And does it impact fitness and aging? And so why this matters, if there's a molecular mechanism, if there's an effect of epigenetic inheritance on organism in nature, and I'm not going to talk about today, but also if there is variation in autocorrelations across the environment, then you have all of the pieces in place where the patterns of autocorrelation in an environment should itself be this selective pressure that either selects for or against transgenerational plasticity to a specific environment. I'm not going to skip this. And so what causes differential gene expression? So we use the same design that we use for the differential gene expression study. Inbred lines, genetically identical, either wounded them, 3 individuals, or did not wound them, grew up the next generation at the second leaf pair stage, collected tissue, extracted DNA, sequenced the methylomes, and then compared the methylation patterns across these 2 different groups. And so I'm not going to go into the details too much, but what we find is that there is abundant differential methylation in this leaf tissue related to whether or not the parents were wounded. We found that most of these regions were relatively small, on the order of about 100 or so base pairs, but you do find some up to about 700 base pairs or so. Average change in methylation across these, about 9,000 or so different sites. You can see most of them was the CHH methylation, and usually about a 22% change in methylation within those regions. So now we want to see, okay, do these overlap with genes, first of all? And the answer is that yes, about 35% of these differential methylated regions do overlap with genes. And when they do overlap with genes, those genes that they overlap with are in fact differentially expressed, or more likely to be differentially expressed than kind of a genome background. And so you really see that for CG methylation, and somewhat for CHG, and significantly for CHH, but really obvious here with CG, where if that gene overlaps with a differentially methylated region, there was about a 12 or 13% chance that that gene, we also previously found in a different grow-up, different experiments, different plants, were differentially expressed, whereas kind of this genome background of about 7 or 8%. Now we can kind of go in and see, okay, what types of genes are differentially methylated. I'm not going to go into this in full detail. You can break it down by, okay, is the coding region of the gene, is that what overlaps with the differentially methylated CG region? So you can break it down by types of methylation, CG, CHG, CHH, and you can also break it down on whether or not it's in the promoter region or in the gene body. When you do that, you find some interesting things, that for a coding region, CG methylation, for example, you find that there's a lot of these nucleotide binding transcription factors that are differentially methylated, things that we think might be involved in kind of these upstream things that kind of lead to different phenotypes. Whereas for CHH methylation and CHG methylation, we see this response to abiotic stimulus show up down here. So I kind of suspect that maybe the CG methylation is what's inherited and what's kind of the upstream factor in this transgenerational response, and that might lead to differential CHH or CHG methylation of more functional genes that themselves are eliciting the phenotypes such as like increased trichrome production or different production of secondary metabolites. Kind of specifically what genes are differentially methylated. So 3 of my favorite kind of candidates that were differentially CG methylated were also found to be differentially expressed and are involved in hormone synthesis, where we got jasmonic acid, ethylene synthesis, and gibberellin. And you can kind of go in there and find that for jasmonic acid and ethylene synthesis, there's differential methylation and increased gene expression, whereas for gibberellin, this 3-beta deoxygenase, there's differential methylation and decreased gene expression. As a whole, you do see that there's trends in the direction of differential methylation and the direction of expression, which is pretty cool. And it kind of goes in, at least for CHH methylation, the way you predict, where we have a lot of genes where there's an increase in methylation and a decrease in expression. And you see it is kind of noisy as a whole, but when you kind of do this contingency table analysis, you find there is this very significant effect where there's more genes than you'd expect that have increase in methylation, decrease in expression. For promoter region or upstream CG methylation, things are a little bit more complex, where you see that when there is an increase in methylation, it's likely there's a change in expression, but it doesn't really always go in one direction or another. And so to get at that a little bit further, we did some modeling, and we found that when there's differences in upstream CG methylation, as well as changes in gene body CHG methylation, it's the interaction between those two types of differential methylation that really predicts the changes in gene expression. So when you have an increase in CG methylation, and also one of these blue lines here, an increase in CHG gene body methylation, those are the genes that are heavily silenced. Whereas when you have an increase in CG methylation and a decrease in the gene body CHG methylation, you tend to find that those genes are actually upregulated. And that was based on 50 or so genes that fell into these categories and also exhibited significant differential expression. A lot of these were transcription factors. In particular, we also found a bunch of zinc finger proteins, which Mariano mentioned, that showed up in his study as often differentially methylated. Lastly, not only is there this directional differential methylation, there's also an increase in the variability of the methylomes in the offspring of wounded plants. So on this one-to-one line right here, we have genes that have equal variance in the offspring of damaged and controlled individuals. And I tried putting these lines, they kind of helped me visualize it. But what you see is that there's a greater density above this one-to-one line here, in both for methylation and expression. And so quickly getting into some of the results as far as field common garden stuff, we want to see, okay, is there natural variation in this response? And so first we found that trichomes do in fact seem to limit herbivory in nature, and that sites with more herbivores also tend to produce more trichomes. Now, what was really exciting is that we found that we used 8 different populations here, and we found that they respond differently to parental wounding as far as trichome production goes. There's a significant effect of parental wounding and population, which shows that some individuals from some areas that the parents were wounded will produce more trichomes, or actually from other areas they do the opposite, produce less trichomes. And when the parents were wounded, those plants actually also tended to be more resistant to herbivory. So over here you can look at, there's not a change in whether or not these green ones were plants that weren't damaged at all, these different sites. But what you see is that if the parents were in control conditions, in this Brad or Ridge Common Garden, for example, there was this pretty high abundance of plants that experienced severe herbivory, whereas when the parents were exposed to damage, you see that really diminished, and that was significant. You also find that there's a population by parental wounding effect on whether or not that parental damage leads to increased resistance in the next generation. So getting back to those questions that kind of started with here, are wound-induced epigenetic modifications transmitted? Yes, there's this complex and abundant differential methylation we find. Do they regulate differential gene expression? It's associated with differential gene expression. We can't yet say that it's the causal factor, though. Evolutionary implications, there is natural variation in this response. And we also see that transgenerational inheritance does, in fact, impact plant resistance in nature.